Hi folks, welcome back. Lecture 18. I want to reiterate something I said previously, not neuroscience at all, which is do let me know if there are stresses, if there are issues, if there's anything that I or the university, honestly anyone might be able to help with in terms of reducing your stress levels, making sure you have all the support you need, making sure the materials are easily available. Effectively, basically anything I or we can do, right? This whole being shut at home, being isolated sucks for everybody. It's probably even worse for folks who are shut in one room or shut in an apartment, or may have family members who are getting sick or needing care. I know certainly folks in my lab, many of the undergrads who've had to go home, are now in the position of caring for their families. They have sick relatives. My point is just, look, if there's something I can do, if there's something you think I can help you get somebody else to help you with, that's part of my job. It's what I would do anyway as a human being. Do let me know. All right, science. Picture on your screen. This is some of the very earliest CNS fossil evidence. So this is an experiment from an organism called Nereus vivens, basically comparing a modern day entombment experiment for an existing species to some fossilized remains found from this Cambrian oithropod, Chengianacaris. And so the idea is that probably this was a spinal cord structure existing way back in the Cambrian area, so obviously many millions of years ago, trying to find similarity between that and present day organisms, and suggesting that probably the formation of a spinal cord type structure happened a really long time ago. Anyway, moving on to more recent stuff. This is definitely more recent. This was last year, and it created all kinds of headlines in the popular press. Bringing people back to life. Okay, first of all, it's pigs, not people. And second, you're not bringing them back to life in terms of we have no evidence that cognitive function was restored, or that if it were a human, the sense of self would be there. What it did do was definitely advance the ability to restore health to tissue, at least on a cellular and molecular level. This was an experiment that basically took, so on the left here, brain structure, 1 hour post-mortem, then looked 10 hours post-mortem, and you can see there's much less metabolic activity, there's a loss of neuronal markers and so on. Perfused with a control solution, nothing helped. And then they had basically an improved solution that allowed them to restore some of the brain function, allowed them to bring at least cells, as far as we can tell, back to life. So this is Nature last year. Feel free to look up the paper. I just think it's a fascinating demonstration of one of the advances that we can make these days. Reminder on this stuff, I've been emailing with Ev, haven't yet got a file that works for me in terms of movie, but this isn't like me hassling you, just reminding you, we'll get that one up as soon as I can. And then this Thursday, the last one, I think, pre-exam is due on the selective erasure of film memory. I'm going to be talking about that paper in the lecture for Thursday, which I'm probably going to post right after this one. And so, ideally you do your report before looking at that, but as with everything else, we'll play it by ear. So last time we were talking about memory and mechanisms that form memories in the brain. The idea that, as you'll see over and over again and from a variety of perspectives, what we think memories are anyway, a particular memory is stored as an engram, is stored as a pattern of changes across many synapses. And so the way that you reactivate a memory is to reactivate all the neurons in that particular engram, in that particular set of synapses and connections that changed and appears to represent a particular memory. When you reactivate enough of the neurons in that particular network, it activates the memory and will then tend to activate all the associated pieces. So you might start out by remembering, the smell of your grandma's perfume. Recalling that will tend to activate neurons that are tightly associated with that, such as how your grandma looks, the fact that she carries an axe. Wait, where did that memory come from? Oh yeah, that memory was created by me in the last class, that grandmas carry axes. And so now that repeated pairing has caused the links between grandma neurons and axe neurons. You had a pre-existing memory of what an axe is, that was one engram, and a pre-existing memory of what a grandma is, that was another engram. And me messing with your heads in terms of creating a gram-mal act memory has made the interaction between those two engrams much, much stronger, so that one of them can now activate the other. Probably it's more effective right now for grandma to activate axe, because I keep talking about grandmas carrying axes. I don't talk quite as much as being axes being a grandma tool. That seems kind of weird. So there's probably actually a slight asymmetry in how those memories are linked. Nevertheless, the basic concept is you have patterns of synapses, patterns of a set of neurons, an engram being activated. That is the biological, the brain representation of a memory. And I'm watching myself on the screen here, wiggling my hands like this. I guess I can see why it might be helpful to have me in the picture as well as the slides. All right. We're going to focus, as I mentioned, effectively exclusively on declarative memories, things that are stored, at least initially, in the hippocampus. We'll talk today about evidence, and it's pretty clear and well accepted, that that engram, the set of neurons that contains a memory, shifts over time to leave the hippocampus and go out to the cortex. For whatever evolutionary reason, my best guess is so that that set of connections in the hippocampus can be reused, can make new memories and different memories, rather than having some limit, everything being jammed up in the hippocampus. We talked about LTP, long-term potentiation, the dominant, certainly leading, not universal. There's still controversy about how exactly this works, how good a model it is of physiological memory formation. But LTP is by far the dominant model for memory formation, for synaptic change, for ways in which synapses can be altered in a manner that persists in the very long term, hours, days, weeks, years, decades. The artificial stimulus we use here, this tetanus in a hippocampal slice, that is definitely not what happens in the real brain. You don't have these almost seizure-like bursts of activity across a particular synapse, but it does appear to activate many of the same mechanisms that are in fact activated by real-life biological memory formation. And so what LTP is a bigger EPSP at a particular synapse for a given stimulus. And so I've said this several times by now. Each one of these little dots is the same size of stimulus pulse. You've got a stimulating electrode in the presynaptic neuron, activated the same magnitude of charge every time. Then you have a recording electrode in the postsynaptic neuron, and it's recording how big the EPSP is. After LTP has been generated, that EPSP is bigger. And that just means when you activate the presynaptic neuron, the postsynaptic neuron is more depolarized. Or in other words, the connection between those two neurons has got stronger. It's being potentiated. A couple of pictures from the text I like, the one I suggest. Remember that the synapses that are being repeatedly activated tend to get stronger, may physically grow, will certainly be potentiated. And the flip side is, if you have very few pairings or no pairings, if this neuron fires sparsely and never causes the postsynaptic neuron to fire, those synapses will be depressed, depotentiated, and in some cases will in fact vanish. You can in fact get pruning, loss of connections. So whatever this particular engram is over here, maybe this is part of your grandma engram, and this is a scimitar neuron. Well, grandmas don't carry scimitars. We know that grandmas carry axes, and so the connection between grandma neurons and scimitar neurons over time will go down to probably zero strength, and in fact may physically vanish. A zero strength synapse still exists. The connection, the two neurons are apposite, that is, they're right next to one another. But a zero strength means there are no AMPA channels. And so when this neuron fires, okay, glutamate's released, but there's nothing on the postsynaptic side to respond to that. There's nothing that gets opened by this neuron firing. So it's a zero strength synapse. As I mentioned, NMDA channels, AMPA channels. AMPA channels are these simple ionotropic glutamate binds, sodium potassium comes in. Very simple channel, creates an EPSP. The NMDA channel is this coincidence detector, is the channel that is only open when both sides of the synapse are active, because you need glutamate from the presynaptic side. You need depolarization on the postsynaptic side to get rid of that magnesium block and now allow calcium in particular to come in and cause stuff to happen. And so again, from the recommended text, just another picture of the same thing. Under normal transmission, So no plasticity, nothing's changing, nothing's being learnt. This is, for instance, when you activate a memory, when you recall something you already know. Glutamate gets released, comes across, it's the AMPA channels that matter. They cause this, postsynaptic cell to be depolarized. This is normal cellular connectivity. And you should remember that underneath the synapse, there are vesicles containing extra AMPA channels. When you're causing LTP, though, this is when the NMDA channel is key. You've got lots of glutamate being released. The postsynaptic cell is also depolarized. And so now calcium comes in and does several things. And in particular, we talked about nitric oxide being released and going back across the synapse to increase the next level of glutamate. And we talked about CAM kinase 2, which both puts these extra AMPA channels into the membrane in kind of the short-term LTP, and finally activates CREB and downstream systems to cause LLTP, to cause permanent changes in the synapse. And so after LTP, what LTP is an increase in AMPA channels, so more receptors for glutamate, and an increase in glutamate being released. Either or both of those two things will make this synapse stronger. This shows you the same thing. This is just the picture I used previously. And so I want you to have multiple ways of thinking about basically the same information. After calcium has come in, and I mentioned, I think previously, if you just inject calcium, if you took this preparation here, and all you did was put some calcium directly into the synapse using a micropipette, that would activate all this downstream stuff. And so to cause LTP, the only necessary and sufficient thing is that glutamate enables some calcium to come in through the NMDA channel. And so if you artificially just inject that calcium, the synapse basically thinks, hey, I'm getting potentiated, and all this downstream stuff will directly happen. So calcium comes in, binds to calmodulin. That calcium calmodulin then activates this CAM kinase 2, this downstream enzyme. And one of the cool things about that is that because it has this auto-inhibitory domain that gets inactivated when calcium calmodulin binds, now each CAM kinase 2 can keep an adjacent CAM kinase 2 turned on, even when calcium has gone away. And so you get this switch that flips into the on state. That synapse is potentiated. And one of the things it does is cause these AMPA channels to go into the membrane. So you make the synapse stronger because of CAM kinase 2, and that state can persist, certainly for hours, until just by random chance, these protein subunits float apart, and then instantly they turn off again. So just getting to this stage, just getting CAM kinase 2 active, isn't going to make a long-term days, weeks, months, years memory. In order to do that, as we said, you've got to activate the whole downstream signaling cascade, including nuclear activation via this transcription factor CREB. The products that are activated by CREB then come back to the synapse and are trapped by some kind of tagging system that potentiates specific synapses. Only those synapses where LTP is happening. Other synapses where nothing's happening, the proteins are not going to stick, they're not going to get trapped. Whatever this mechanism is, it only happens at the specific LTP synapses. My usual, slightly emotionally sad moment where there are no questions because you guys aren't here. I will note that in a normal timeline semester, this is where you would get a pop quiz. So you can have the anxiety of having a pop quiz and then realize, oh, there's no pop quiz. But instead, I have to ask questions online. So I'm really glad you guys are doing that. I'm enjoying seeing them. I'm looking forward to responding to them. I think it's a nice extra way to interact. One thing we haven't talked about, and are now going to do, is activation of a memory. What happens and how do those memories change? And I'm going to go back to my example of grandmas and exes. You had a memory of grandma before this class. Some of you maybe didn't if you didn't have a grandma, but even then you had a memory for what the concept of a grandma was, what the word grandma meant to you. That memory has now been altered by the inclusion of axes, right? How do memories get changed? Once you've formed a memory, how do you alter that memory? How do you improve it? How do you refine it? How does that memory? So that process we call reconsolidation. Consolidation is where a memory gets put into LTP. Reconsolidation is taking an existing memory, tweaking it slightly, and putting it back, reconsolidating. So I talked last time about the fact that you probably need this Krebs activation for long-term LTP. What about for reconsolidation? What about for making a memory slightly different? So what you're seeing on the screen is one of the most impactful papers in this field in the past couple of decades. This is from 2002, so it's almost 20 years old now. It's still a interesting paper. Cristina Alvarini is still one of the leading figures in this field. In fact, one of my graduate students is working with her for his thesis. Here's what's happening. The canonical figure from all this stuff is here. And what it's showing you is the consequences of giving anisomycin. Anisomycin is a protein synthesis inhibitor. It's a drug that blocks protein synthesis. Let me take you through the bars here quite slowly. On the y-axis is the latency to enter a dark compartment. If you take a rat and you put it into an apparatus called an inhibitory avoidance apparatus, it has a very brightly lit area and a door and a very dim area. If you put the rat in the bright light, lower the door, the rat will run into the dark. Rat prefers to be in the dark quite spontaneously and rapidly. What you can see is the mean latency to enter the dark here on the initial trial, what we call the acquisition trial, is very, very low. And I've done this task, probably thousands of times, certainly hundreds of times. It's very reproducible. You put a rat in a bright light compartment, you open the door to the dark, rat runs in. What then happens is as soon as the rat enters the dark, it gets an electric shock into its feet. It's called a foot shock. The apparatus has a metal floor. You activate a current, you give the rat a shock. You pick the rat up, take it away, put it back in its home cage. What has the rat learned? The rat's learned, hey, that dark is scary. It isn't this nice, comforting, dark place I thought it was. It's actually kind of scary. You've made a fear memory. So you bring them back the next day. Test day number 1. You put them in the light compartment, you open the door, and what you see is that now the latency to enter that dark has massively increased from effectively 0 up to about 9–10 minutes-ish. And usually there's a ceiling, a cutoff of 10 minutes. So most of these rats are taking a much, much, much longer time to go into the dark. That was their default yesterday, but today they've learned and they clearly can activate this memory. Hey, wait, dark, scary, that particular location, I'm gonna get hurt. I'm not going in there. That part's easy, right? And so in the vehicle condition, that memory is intact. If the first test day is accompanied by administration of anisomycin. So you've taken this rat, you've made a memory on the acquisition day. You've activated that memory, and now the rat says, hey, no, that's scary over there, I'm not going in the dark. If when that memory has been activated, you give this anisomycin drug, blocks protein synthesis, what you see is that you bring them back on day 3, so a further day later. That memory appears to have been effectively eliminated. The latency to go into the dark is now very, very short, much shorter than on day 2. In contrast, if all you gave the rat on this first test day was a saline, was a vehicle, then you bring them back the next day, they're still very afraid of the dark. So the basic interpretation here, which turns out to be correct, is that by giving anisomycin at the same time as you've activated this memory, you've made that memory go away. You've wiped it out. You've basically prevented reconsolidation. A really important control is this gray bar. These are animals where the memory was created, and then on day 1, nothing happens to them. That memory isn't tested. They are only given anisomycin. So they're given the same drug as these guys, but it's not paired with a memory. There's no memory reconsolidation taking place. And what you can see is that when they are brought back on effectively day 2, the same time as these guys, that memory is still there. It's clearly impaired. Giving anisomycin clearly did something that was not great for the memory, but it's equally clearly not this wipeout of the memory that you see in the group where the anisomycin was given at the same time as the memory was activated. So this experiment was, widely interpreted as evidence that protein synthesis was critical, was essential for the reconsolidation of a memory, for that memory to be put back. And our basic understanding of memory, I think, not very controversially across neuroscientists, is that a memory long-term storage is kind of like the long-term storage of a computer in some ways. The file sits there, nothing happens. When you activate it and recall it, you basically bring it back into short-term memory where it is vulnerable. The weird thing is that unlike a computer where that file still also exists in long-term memory, effectively what seems to happen is you bring the only copy of the memory up into the short term where it is now vulnerable. And if you interfere with the restorage, that memory goes away. The file gets lost rather than just going back to its previous state. So if you want to wipe out a memory, what these guys showed was, well, give the rat some anisomycin when the memory is active, and that memory will go away. We're going to talk about variations on this experiment for a while. And in particular, this is the second of the two papers that you can write a critique of. So the first was McNay, 2000. The second is this paper by Canal. Clint Canal was a graduate student. I did some of his training. Paul Gold is obviously my doctoral advisor. This is one of my favorite papers in neuroscience because it's a really excellent example of identifying a potential confound, doing a set of experiments that test that confound and saying, yeah, those previous data, they weren't wrong, but they don't mean what you think they meant. So here's what they did. This is the summary slide, and we'll go through it more slowly after this. But they basically did the same experiment, reproduced Alberini's work, showed that yes, if you give anisomycin, which is abbreviated ANI here, and paired it with a pre-existing memory, you definitely did create amnesia. Then they showed that they could do a couple of things to reverse that amnesia. And lastly, they showed that just giving norepinephrine by itself, so a neurotransmitter that had no effect on protein synthesis, could have that same amnesia effect. All right, that's the summary. Walk through it more slowly. So here's the abstract, right? The canonical theory in the field is that there are strong study data saying, hey, you need protein synthesis to restore memories. Giving anisomycin, their point was, might do something else that isn't just blocking protein synthesis. What if anisomycin is doing some other thing, some side effect, and it's that side effect that is actually causing the impairment to reconsolidation? So they used microdialysis. And what they saw was that when they gave anisomycin into the amygdala, so into a modulation site for the memory, what they saw, as well as this impairment, this amnesia, was massive increases in several neurotransmitters. Norepinephrine in particular, also dopamine, serotonin, thousands of % higher than baseline. For comparison, when I do experiments and I'm measuring changes in neurotransmitters during normal brain activity, if you see a change of 50%, it's pretty big. 100% is massive. So changes in the thousands of %, this is abnormal. This is a wave of neurotransmitters that's probably causing seizures, that's certainly having massively abnormal effects on the brain structure. So there definitely is a side effect. If they blocked the impact of that side effect, so they gave anisomycin, they also gave drugs that manipulated norepinephrine activity directly. So they manipulated the beta-adrenergic receptor, which is the receptor in the brain for norepinephrine. And they either blocked it or boosted it at a slightly different time. That blockade of the beta-adrenergic receptor prevented the amnesia. Protein synthesis was still blocked. Anisomycin was still doing what it was intended to do, but the side effect, this massive release of norepinephrine, was being blunted or prevented, and the amnesia was attenuated. And lastly, if they just gave norepinephrine without giving any anisomycin, so protein synthesis wasn't affected at all, the norepinephrine by itself was enough to cause amnesia. And so their conclusion was, and you guys will see this when you read the paper, you really should go read the paper. Papers that I think are my favorites are much more likely to come up in exams, right? Anyway, their conclusion was the anisomycin studies that had been held up as strong evidence for a requirement of protein synthesis in reconsolidation really couldn't be interpreted that way, because this side effect confound was so big and could, by itself, entirely explain the effects of the experiment. Here are the figures from the paper. So figure 1 is just, does protein synthesis actually get inhibited? CFOS, we talked about this briefly, is an immediate early gene. It marks cells that are very active. And so you can see in the amygdala, which is part of the site where fear memories are made, animals that have this inhibitory avoidance task definitely show an increase in CFOS, in protein synthesis. Anisomycin prevents that, shown in the graph here too. So anisomycin is doing what it's supposed to do. It is preventing protein synthesis. So far, so good. Here are the microanalysis data. And this is the kind of gross scale. Here's the close-up. The close-up is needed so that you can see that even in animals that have vehicle, there are small increases in these neurotransmitters. As I said, 100% is a big effect. And these guys are getting increases in norepinephrine and dopamine, and maybe even a bigger increase in serotonin, on the order of 100% or so, when the rat is doing this inhibitor avoidance task. So the amygdala is clearly very active. It's very involved in processing this task, as you would expect. But that vehicle line is barely above the baseline of the graph on this other scale, because these huge increases, when you give anisomycin, just dwarf it, 2,000, 5,000, many thousands of % increase in these neurotransmitters. Just obviously much more dramatic than anything you see in the absence of anisomycin. All right, so propranolol is a beta-adrenergic antagonist. And when they gave propranolol, as well as anisomycin, it attenuated that amnesia. Clenbuterol is actually an agonist. And the reason they gave clenbuterol is because if you look closely at this norepinephrine graph, you can see that in the anisomycin group, later on in the experiment, norepinephrine was actually way below baseline. And effectively what I think had happened is all of the norepinephrine had been used up. The neurons had been so overstimulated, they'd released all their norepinephrine. And so a little later in the experiment, they just didn't have any left. So clenbuterol was given at this time point to boost norepinephrine back up towards baseline. Both these treatments then, an antagonist during that huge spike, and an agonist during the below baseline levels later on. Both those were given to correct the norepinephrine signaling back towards that seen in control animals. And you can see that both those treatments didn't completely restore memory, but they certainly attenuated the massive amnesia you're getting down here. Also, notice the y-axis, it tops out at 180 seconds. This is a pretty short memory span, right? So the rat is just saying, I'm just going to stay in this light compartment for 3 minutes, as opposed to maybe a more usual 10 minutes or so. Honest, I'm not quite sure why Clint chose to use such a short time span, but certainly you can see that attenuation of amnesia was pretty effective. Last bar here. If instead of giving any anisomycin, Clint just gave a dose of norepinephrine into the amygdala that was the same as the dose created by anisomycin, you can see it looks just the same. The amnesia was just as intense, but protein synthesis is still entirely intact. So, you can read this for yourself, blah, blah, blah. Basically what we're saying is, hey guys, you didn't think about possible side effects, and it turns out probably your conclusions were entirely unjustified. There's this whole side effect of neurotransmitter release that really says your evidence for protein synthesis being required for reconsolidation, for restoring of this long-term memory, just isn't there. Go find a different method. And the reality is no one has. I think everyone still believes that protein synthesis is probably still important and required, not just for initial memory formation, but also for this restorage. It's just that the data aren't that good now. There isn't a good tool that's been shown to be confound-free. All right. That was, I suppose, a slight diversion into memory reconsolidation, into how memory gets changed. Because that reconsolidation allows you to change the engram slightly, allows you to add new information, not just that grandmas wear pink and like fluffy sofas and make cookies, but also that they carry axes, right? That memory has been reconsolidated and altered slightly. We'll look at that again in a minute. Before we do, I'm going to go back to the basic mechanisms of initial memory formation and just run through some of the evidence for the whole cascade we've been talking about. The NMDA channel, going to calcium entry, activating CAM kinase 1, the calcium coming through that NMDA channel and causing AMPA subunits to go up to the membrane, yada, 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 all those different steps. Many of these tasks have used a maze or a spatial memory task. We call the Morris water maze task, named after a guy called Richard Morris. He didn't invent it. It was invented by Carol Barnes. She didn't call it the Barnes maze. She just called it the water maze task. Richard Morris then named it the Morris water maze task. He's kind of a dick. The canonical experiment goes something like this. Here's a water maze. And when you're actually doing an experiment, it's filled with a little bit of powdered milk to make it opaque. Just underneath the surface of the water, there's an escape platform. And then you're doing this in a room full of visual cues. So you put the rat into the water tank, doesn't like that, it wants to escape, swims around the tank until it finds the platform. Gets up on the platform, the experimenter takes her out, dries it off, puts it back in its cage. Over time, and in fact fairly rapidly, the rat learns, here's the room, the escape platform is always over here in this one location. And so the amount of time it takes for the rat to escape goes down and down and down. Instead of being wandering around the tank aimlessly, kind of looking for some hope of escape, the rat says, okay, you put me in here, that means the platform's over there, zoom, and goes straight to the platform. If you want to manipulate the NMDA channel, you use an ICV, an intracerebral ventricular, so into the ventricles administration, of that APV drug we've seen previously, this drug that blocks the NMDA channel. You can also put it directly in the hippocampus. You could probably put it into the amygdala because this is a somewhat stressful task. It's cold water, it's kind of aversive. But in general, we're looking at hippocampal activity. So you can either put it in ICV, where it will affect the whole brain, or you can administer this drug directly into the hippocampus. When you do that, as we've seen previously, you get much less or no LTP generated. Dentate gyrus is part of the hippocampus. And if you look at the performance of these two groups, the control animals here in red pretty rapidly decrease their escape latency, the time it takes them to get out, down to below 10 seconds. It really is. Get in, look around, oh yeah, okay, platform's over there, swim. The animals that have the NMDA channels blocked, they can learn it a bit, they learn better than their first baseline, but they're never very good at it. They never form a particularly strong memory representation. Even more clearly, if you now take the platform out, put the rats in, and see where they search. So you've got the target quadrant, that's where the platform should be, opposite to adjacent. A control animal will spend most of their time looking in the target quadrant. They've learned where this platform is, now they swim around over here saying, hey, dudes, where's the platform? It was here. An animal where the NMDA channel was blocked shows no preference. So although they got better at the task, maybe they just learned to swim better or something, they show no evidence for a memory of where that platform should be. They're still just swimming around the maze, unable to say, hey, we know where the platform should be. So this is called a probe trial. You're probing the memory by taking out the platform and allowing the rat to look around. All right, so that's the basic experiment. What if you just mess with the NMDA channel? So the NMDA channel is made up of 4 protein subunits. This is the whole channel sitting in the membrane. Every NMDA channel has 2 NR1 subunits, and then 2 NR2s, and the NR2 subunits come in 2 types, 2A, 2B. Insert your own Shakespeare joke here. It turns out that 2As and 2Bs are slightly different. We'll come back to that. If you genetically just knock out the NR1 subunit, you can do that in a certain set of cells. This uses methods like the Crelox tool we saw previously. And so Suzumi Tonegawa, who's at MIT, and is also a bit of a dick, honestly, did this great experiment. He has a really world-class memory lab. He knocked out the NR1 subunits just in CA1 neurons, just in this one part of the hippocampus. Here's the control animals. You can see it's a very nice dark band of cell bodies. Here's the knockout animals, and you can see they're missing the vast majority of these NR1 subunits in the CA1. When you do that, what you see is they can't make LTP in this brain region. Right next door, in the dentate gyrus, so just to give you anatomy, this is the CA region, CA1, CA3, and so on. The dentate gyrus is this bunch of neurons down here. The whole thing is a slice through the hippocampus. In the dentate gyrus of these animals, LTP is perfectly intact. So the brain area right next door, where they have the NR1 subunits, everything is fine. It's only impaired in CA1. And it turns out that just knocking out NR1 subunits in that CA1 bit of the hippocampus, again, you see this learning impairment, and on a probe trial, animals show no memory. So NMDA channels, specifically in the CA1 area of the hippocampus, appear to be critically required for spatial memory formation. I said I come back to the NR2s, 2As and 2Bs. It turns out that channels that have the 2B subunit stay open a little longer. They let a little more calcium through. And what that means, in fact, is that it is more rapid to form a memory if you have a lot of 2B subunits. So Joe Sien, who's not a dick, he's a perfectly nice guy, made this mouse, which he called a Doogie mouse, after Doogie Howser, MD. And if you don't know who that is, ask your parents. They will explain to you why this is funny. Anyway, he made a mouse called the Doogie mouse. And this mouse was very rapidly able to form memories, much better than an average control mouse. So here's the LTP. You can see LTP is more rapid and much, much higher in these NR2B overexpressing mice. And their memory is also better. This is a slightly different task. This is a freezing task. Basically, you teach the mouse to be afraid of something, and a mouse's response to being afraid is just freeze. They're prey animals, that's their default response to kind of threats. And so you can measure how much of the time they freeze. These mice with more NR2B subunits froze more, showed better memory over even up to 10 days later. So the NR2B subunit appears to promote memory. And in fact, there's some evidence for that in the wild too. If you look at songbirds, which have a critical period during which they're required to learn a very complex mating song, specifically during that period, they get extra NR2Bs in their hippocampus. And so it's basically set up to be a very rapid learning point in the life of the songbird. And then when you go back to not being this critical period, the NR2Bs go back to baseline. We humans have more NR2Bs early on in life. They decrease a little later in life. So we are better able to learn things early in life. Those memories, kind of side effect, become more resilient to some extent later in life because they're less able to be altered. During early life, we learn new things easily. We also forget things more easily and change our memories more easily. As you get older, you're less able to learn new things, but the memories you do have become more resilient. And part of that is because you lose these NR2B subunits. All right, so NMDA channel, check, appears to be important. What about AMPA channels? You know, remember the concept. You've got these AMPA channels hanging out inside the neuron, underneath the membrane, and they go up to the membrane during the process of making the synapse stronger, during the process of LTP. Rob Malinow, perfectly nice guy, measured how many AMPA channels were at the cell surface of membrane by basically expressing a fluorescent tag that only showed when the tagged AMPA channel were at the cell surface. And what he did is an experiment I still find quite remarkable, which is he gave two groups of rats exactly the same number of foot shocks in the same environment. Some of them, those foot shocks were paired with a tone or a light, basically a cue that allowed the rat to form a memory. Oh, when I hear this tone, I should be afraid it's going to hurt. The other rats got the same number of shocks, but they weren't paired with anything particular. It was just, we're getting foot shocks. Ow, that hurts. And what he saw was that the number of AMPA channels and the percent freezing increased together. And so, unsurprisingly perhaps, when they played the tone, you know, the animals that had these paired shocks, every time they got a tone, they had a foot shock, they showed much more fear. They froze much more. Whereas the animals that had been having foot shocks just kind of randomly had no particular reason to be afraid of a tone. And then when you look at the synapse level in the membrane, what you see is that the animals that had these paired foot shocks have many more AMPA channels going up to the cell surface. If you look at the LTP, it was also stronger in those guys. The flip side is, if you give a modified gene for the AMPA channel, so that you make it non-functional, you insert a copy of this AMPA channel gene that creates a non-functional AMPA channel, and you can tag it with the green fluorescent tag so you can see where it is. Then what you see is that the initial LTP is okay, but it decays very rapidly. Those AMPA channels don't hang around. They don't continue to allow, greater EPSPs. Basically, the LTP decays on the order of 10, 20 minutes, whereas a control animal, it persists for a long time. And that is matched by an impairment in memory. So the animals that have received, it's actually a viral vector. You basically create an artificial virus that contains the gene for this non-functional AMPA channel. You inject it into the hippocampus. And what you see is that animals with that injection have greatly impaired memory. Messing with the AMPA channel impairs the memory. Okay, NMDA channels, AMPA channels. So far, so good. Also, if you give one of a class of drugs called ampakines, and these are actually on the market, there's a company that I think is called Memory Pharmaceuticals that makes ampakines. You can study sign up for clinical trials and so on. And what they do is they bind to the AMPA channel and keep it open for longer. It's basically like having extra NR2B subunits for the NMDA channel. But in this case, it's working with the AMPA channel. It's keeping that AMPA channel open more. And so it's allowing more EPSPs for the same amount of glutamate. What you see is that animals treated with that drug learn more rapidly. They get to the same level of learning in the end as control animals, but they pick up new information more rapidly. There are some downsides. They also forget things more rapidly, but certainly this class of drug has been now tested for memory impairment conditions, Alzheimer's, mild cognitive impairment, aging, that kind of stuff. Two different types of channel, NMDA, AMPA. As the blurb here says, what we've learned is that NMDA channels are critically important for learning new stuff, for creating LTP. AMPA channels, well, you need AMPA channels to allow you to get LTP in the first place, because you've got to depolarize that synapse for the NMDA channels to be able to work. And then when you retrieve a memory, that's all AMPA channels. That's just normal transmission across the synapse. So you'd expect AMPA channels to be important both for learning and then for retrieval, for showing that you have that memory. And that's exactly what you see. So here, this particular task, the rat is given a series of food wells, and they're sometimes covered with different objects. The rat has to remember which objects basically are covering the food well. Control animals can do this just fine. APV, this is the drug we've seen before, blocks NMDA channels. If you give it when they're learning, then you markedly impair their memory. But if you give it when they come back to show you whether they have a memory, it has no effect. The memory's already there. You don't need NMDA channels to retrieve an existing memory. You only need them to create new memories. In contrast, CNQX, another long chemical name, you don't care what it stands for. What it does is block AMPA channels. So APV blocks NMDA channels, CNQX blocks AMPA channels. If you give that when they're trying to learn the task, nope, can't do it, they're impaired. But also, if you give it when they're trying to show you that memory, when they're trying to perform this memory task later on, they're still impaired. So you need the AMPA channels open both for learning, for creating that LTP, and also later on for normal synapses to activate the memory to use it, to say, oh yeah, the food's over here. AMPA channels, just once again to reiterate, needed for learning, needed for retrieval. NMDA channels, needed for learning, not needed for retrieval. Once the memory is there, you don't need the NMDA channel anymore. The memory will persist without any further need. You can do similar experiments for all the steps in this molecular cascade we've been talking about. So if you create mice where you knock out CAM kinase 2, you can look at the figure yourself. You see the pattern of data you'd expect. Can't learn the task as well, especially if the platform is hidden. They're perfectly fine at recognizing, oh yeah, there's a platform over there, and swimming to it. Their swimming isn't impaired. But their ability to remember where a platform is markedly impaired, confirmed by this control trial. If you mess with CAM kinase 2's autophosphorylation ability, that ability to keep the other CAM kinase 2s turned on, then what you see is it takes much more learning, many more trials to learn the same amount of memory. You can learn it. And the reason you can learn it is because if you learn things strongly enough to activate CREB, you no longer need CAM kinase 2. You've gone past that stage of SLTP, and you're now into LLTP. So if you teach the animal, I think it's mice here, could be rats, doesn't really matter. If you teach the animal the same thing enough times, you'll create a strong enough association, a strong enough pairing to activate CREB and create LLTP. But the initial learning, this kind of SLTP stuff, that requires CAM kinase 2 and this autophosphorylation, this switch flipping. If you mess with just that function, you cause this impairment in rapid learning. It takes much longer to activate this LLTP. Same thing here. So the evidence for every step we've been talking about, everything from initial activation of NMDA channels to calcium entry, to CAM kinase 2, to AMPA channels going up to the surface, there are really good data for all those steps. It really is a case where we can now be fairly confident that we understand the molecular biology of exactly how this, very psychological memory thing, a memory, of exactly how that memory is being created at the molecular level. Where is it happening? So this is obviously the paper. There'll be a class presentation as soon as I can get a video up and posted, looking at where that memory is stored. And initially, we've been talking about the hippocampus. That's completely correct. The initial engram, the initial set of synapses for any declarative memory is in the hippocampus or the medial temporal lobe. And this is another example of, of where we learned from HM. So I'm going to let this video play for a second.
On September 1st, 1953, William Scoville used a hand crank and a cheap drill saw to bore into a young man's skull, cutting away vital pieces of his brain and sucking them out through a metal tube.
Meanwhile, I will get coffee.
This wasn't a scene from a horror film or a gruesome police report. Dr. Scoville was one of the most renowned neurosurgeons of his time, and the young man was Henry Molaison, the famous patient known as HM, whose case provided amazing insights into how our brains work. As a boy, Henry had cracked his skull in an accident and soon began having seizures, blacking out, and losing control of bodily functions. After enduring years of frequent episodes and even dropping out of high school, the desperate young man had turned to Dr. Scoville, a daredevil known for risky surgeries. Partial lobotomies had been used for decades to treat mental patients, based on the notion that mental functions were strictly localized to corresponding brain areas. Having successfully used them to reduce seizures in psychotics, Scoville decided to remove HM's hippocampus, a part of the limbic system that was associated with emotion, but whose function was unknown. At first glance, the operation had succeeded. HM's seizures virtually disappeared, with no change in personality, and his IQ even improved. But there was one problem. His memory was shot. Besides losing most of his memories from the previous decade, HM was unable to form new ones, forgetting what day it was, repeating comments, and even eating multiple meals in a row. When Scoville informed another expert, Wilder Penfield, of the results, he sent a PhD student named Brenda Milner to study HM at his parents' home, where he now spent his days doing odd chores and watching classic movies for the first time, over and over. What she discovered through a series of tests and interviews didn't just contribute greatly to the study of memory, it redefined what memory even meant. One of Milner's findings shed light on the obvious fact that although HM couldn't form new memories, he still retained information long enough from moment to moment to finish a sentence or find the bathroom. When Milner gave him a random number, he managed to remember it for 15 minutes by repeating it to himself constantly. But only 5 minutes later, he forgot the test had even taken place. Neuroscientists had thought of memory as monolithic, all of it essentially the same, and stored throughout the brain. Milner's results were not only the first clue for the now familiar distinction between short-term and long-term memory, but showed that each uses different brain regions. We now know that memory formation involves several steps. After immediate sensory data is temporarily transcribed by neurons in the cortex, it travels to the hippocampus, where special proteins work to strengthen the cortical synaptic connections. If the experience was strong enough, or we recall it periodically in the first few days, the hippocampus then transfers the memory back to the cortex for permanent storage. HM's mind could form the initial impressions, but without a hippocampus to perform this memory consolidation, they eroded like messages scrawled in sand. But this was not the only memory distinction Milner found. In a now famous experiment, she asked HM to trace a third star in the narrow space between the outlines of two concentric ones, while he could only see his paper and pencil through a mirror. Like anyone else performing such an awkward task for the first time, he did horribly. But surprisingly, he improved over repeated trials, even though he had no memory of previous attempts. His unconscious motor centers remembered what the conscious mind had forgotten. What Milner had discovered was that the declarative memory of names, dates, and facts is different from the procedural memory of riding a bicycle or signing your name. And we now know that procedural memory relies more on the basal ganglia and cerebellum, structures that were intact in HM's brain. This distinction between knowing that and knowing how has underpinned all memory research since. HM died at the age of 82 after a mostly peaceful life in a nursing home. Over the years, he had been examined by more than 100 neuroscientists, making his the most studied mind in history. Upon his death, his brain was preserved and scanned before being cut into over 2,000 individual slices and photographed to form a digital map down to the level of individual neurons, all in a live broadcast watched by 400,000 people. Though HM spent most of his life forgetting things, he and his contributions to our understanding of memory will be remembered for generations to come.
So with luck, you're getting a sense of just how critical HM was and still remains to our study and our understanding of different memory types. So you may have noticed minor tragedy out of coffee, so I'm going with milk. The Smith and Squire paper basically tried to confirm some of that insight into where memories were stored. And so what they were looking at was activation of the hippocampus and the temporal lobes versus the cortex for memories of different ages. The first experiment was in patients that had hippocampal lesions. And so in control patients, recent whacks on the head and causes of amnesia, for instance, more recent memories activated the hippocampus, less recent memories, much less so. It's not zero. When you use your hippocampus, you're probably still using it as an index to where memories are stored and so on. But what was interesting there was for these patients with hippocampal lesions, recent memories hardly activated the cortex at all, hardly activated the hippocampus at all. What they saw was a much greater ability to restore memories from longer ago. So normal people, you and I, are better at storing memories and recalling memories from more recent times, and we get more activation of the hippocampus. Patients with hippocampal lesions of varying amounts. I mean, obviously in some cases, there's no hippocampal activation at all. And the key point here is that you do worse on memory for recent events, so things that happened around the time of your injury perhaps, and much better for more distant events. So something that happened 25 years before your injury is recalled as well or better than control patients. But things that are recent, things that maybe happened up to 5 years before the injury that caused you hippocampal damage, you're much worse at recalling those, in contrast to healthy people without brain injury who are much better at recalling those more recent things. So this psychological observation suggested, remote memories, things that happened a long time ago, don't appear to be sensitive to hippocampal damage. But more recent memories, and that more recent on the scale of years, this is the 5-year time point, right? Memories 5 years before you got hippocampal injury are still impaired compared to people without hippocampal injury. So then these guys at Smith and Squire looked at activation of different brain structures by memories of different ages. So memories for things long ago, 30 years ago, memories for things in the recent day. And what they saw for the hippocampus is exactly what they would have predicted. That is greater activation in either the left hippocampus or the combined left hippocampus amygdala for more recent events, memories from maybe 3 years ago, less so for much more remote memories. And in contrast, if you look at some of the cortical areas, you see the opposite. So less activation for recent memories, but an increased amount of activation across several different cortical areas, right? An increased amount of activation for more remote memories. So things 30 years ago causing a greater activation compared to things more recently. The basic conclusion from this, and it's borne out by brain lesion data and by scanning data and by a variety of sources, is basically that we've seen this engram, this point at where the memory is stored, move. For more recent memories, and recent really is, maybe a year, 2 years, 5 years, there's a strong component of hippocampal storage. As that memory is persistent and is maintained and maybe is reactivated a couple of times, it slowly moves out to be stored more in a cortical network. So it's less vulnerable to hippocampal damage. Think about HM. He didn't forget his name. He didn't forget his mother. He didn't forget the street he grew up on. All that stuff was long before his hippocampus was removed. What he lost was some small amount of time before the operation and everything going forwards. So when we talk about the engram, the set of synapses that represent a memory, that engram moves. And it starts out, calling on the senses that are sensing this thing we're going to remember, activating the hippocampus, forming a set of connections. And then over time, that set of connections changes to be now using more cortical neurons, fewer hippocampal neurons. Slight change in topic. We'll come back to this next time. We've seen mechanisms involved in storing a memory. What about removing a memory? Can we, for instance, take advantage of some of that stuff to do with reconsolidation and anisomycin to go in and take out a particular memory? And you guys should be able to think of ways that would be really useful. PTSD. What about addiction? Many of us can think of memories we might prefer not to have. Can we go in and selectively delete your memory for spiders if you have a spider phobia? So, as I said, this is where we'll start again next time. This again is a paper that's assigned full class and is a really cool paper. And in fact, over the past, probably 7 or 8 years, there have been some really cool advances in this particular field of how to mess with existing memories. And what I'll actually show you is how to implant completely false memories, memories for things that didn't happen, but nonetheless can be created in the mind, in this case of a rat or mouse, that will then look just like an accurate real memory for a real event. Thanks. I need to get coffee, obviously, and it's 55 minutes. So we'll leave it there. We'll come back in a second, next class, and talk about some of this erasure of memories. See you then.